Okay, so we're back. We're talking about the cell division here, just to get people caught up and do this really quickly. We will be dissecting the flatworms tomorrow, and we have our hypothetical flatworm bisected longitudinally, and we then have a close-up of the cells along this ragged edge here where he's got to grow back. And then we sort of are looking at the idea that what's going to happen with this cell is these cells are going to have to divide to replace the rest of this critter with new cells so that it, not a he, most flatworms represent both sexes and are therefore hermaphroditic. The flatworm will make exact copies of its cells. In a process called mitosis, which is asexual, and it's just reproduction by cell division, only exact copies of the cells are made. So every single piece of information in this DNA that's activated in this cell will be present in both of the new cells. That's the goal of mitosis. That's the goal of mitosis. The goal of mitosis is to make exact copies. When we talk about sexual reproduction later, in which sperm and egg are made, to put it very bluntly or without any fine points, half the DNA of a normal cell, so that when the sperm and egg reunite, then you'll have the full complement of chromosomes. We'll get into more detail about that a little bit later. But anyway, so what I want to do is I want to take you through the stages of mitosis so you can understand why each one happens. You do not have to memorize these, which is good. In years past, we made people memorize these, but we've sort of made a decision based on the new standards that the more important part is understanding why these two processes are different. What you're going to see is that mitosis, asexual reproduction, and meiosis, sexual reproduction, are similar but also very different. And what we want you to know is why. So what I'm going to do is I'm going to give you the 4, 2, 3, and then I guess we'll have to go over here for the third one. 4 stages that lead to our 2 new cells. And we're going to, so here's the cell beforehand. I'll number these. So this is 1. We'll call this interphase. We've come to the end of interphase. This is not part of mitosis. It's part of the part of the cell cycle called interphase. You got your stages of mitosis here. So here we are in interphase, and we're an organism with 4 chromosomes. And so I'm going to sort of write those in here. You got a red one, you got a green one, you got a blue one, and you got your purple one here.
You want to draw this?
I do. Yes. So this is interphase, right? Interphase is just a cell being a cell, having its regular life. And the cell is about to go through the stages of mitosis. We will say they go in this order. 1, 2, 3, 4, 5, which is sort of this whole thing here. It's not as bad as all that. Honestly, folks, I have to tell you. It all sort of makes sense. And what you need to understand is why this happens in terms of protecting the DNA. Because, I mean, what happens if this thing makes 2 copies and they don't have exactly the same DNA? What's bad, right? Yes, not good. Bad things happen. If you end up dividing the cells and all the chromosomes, or half the chromosomes go on this side, half the chromosomes go on this side into the new cells, that's good. But if like a little piece of one gets tangled in the other, that's bad. So all the things that happen are really there to keep bad things from happening. First thing that happens in prophase, well, 3 things happen in prophase, actually. And the first one is that the nucleus dissolves. It disappears. Nucleus disappears, and so we had a nucleus here, but now it disappears, and that gives, why is that? It's because it gives more room for the whole thing to happen, so it's not like trying to do all this stuff in a tiny little space. Instead, they're like, okay, we're going to dissolve the nucleus, we're going to give you all this cell space to do your cell division here. And so then the other thing that happens is that the chromosomes wind up. And this goes back to our Christmas lights analogy, where you don't want to have it like it was before, right? The nucleus before was just a mixture of different threads of chromatin, and they're all bundled up in there together. And so this is no way to do moving day, right? Especially if you're going to two different places. And don't forget, at the end of interphase, what have all of these lengths of chromatin done by the end of interphase? What do they do though? What is, like, if I had like the red chromosome, the red stretch of DNA here, what is that going to do by the end of interphase?
Do they get copied?
They get copied, that's right, thank you. They make a copy of themselves. So each of them, remember, makes a little copy. It's connected by a piece of protein called a centromere. And so actually at the end of interphase, what you really have is two copies of every one attached by a centromere. Each of these eventually is one will go in each new cell. That's the idea. That's why they do that, is so that in the new cell there can be, two of everything. So those will split up that way. But before that, they have to wind up into something a lot more manageable. So they supercoil. I showed you a diagram of that. They wind around these little proteins called histones, and then those stretches wind around themselves, and then those wind around themselves, until you take this 6-foot long thing and you turn it into a little tiny coil that fits very easily inside the nucleus without being all spread out all over the place. And so all the chromosomes are going to wind up in prophase, and we end up with 4 of these little structures, which are not called chromosomes at this point. They are called something else. They are called sister chromatids, because why wouldn't we name something similar but dissimilar so that it's harder to remember? So yep, they're not chromosomes, but each of these 2 copies is called a sister chromatid. And they're exact copies of each other, don't forget. All right, so the third thing that happens, and there's only 3, is that the spindle forms, and the centrioles migrate to the poles. What does that mean? Well, the centrioles are these 2 organelles that hang out near the nucleus. And when the time comes for cell division, they migrate to the poles. So one goes to one extreme end and the other goes to another extreme end, and they create spindle fibers, which are these fibers, these protein fibers. The spindle fibers are here to move the chromosomes around the nucleus as they need to be. They control them through this whole process again. So why is this important? Nucleus disappears to make more room. The chromosomes wind up so they don't get tangled in each other. Using the Christmas lights analogy, you don't want to have a giant box of Christmas lights that are totally unwound because you can't separate them. So if they're all wound up on some sort of thing, piece of cardboard or whatever individually, then it's easier to move them around. And that's sort of like what this is. And then the spindles form, and that's to control the movements of the chromosomes. So those are the three things that happen in prophase. The other ones are a lot simpler. In the next one's called metaphase, and what happens is that the chromosomes line up on the equator of the cell, moved by the spindle fibers. So now it sort of makes a little more sense why the spindle fibers are there. They have moved them so that chromosomes line up at the equator. And why do they do that? Again, because they're soon going to get pulled apart. That's what happens in the next phase. Let's talk about both of these together. What happens is they get reeled in by the cell. Whoops, I switched. No, I didn't switch them. And so these go together in a really important way. They line up along the middle, because if they're lined up along the middle, they're not likely to get tangled in each other. When they get pulled apart, they're just going to go back to each side, because that's the goal. Remember, they're going to split eventually, and the idea is that we want 4 chromosomes here and 4 chromosomes here, no tangled broken pieces, no extra chromosomes, anything like that. And so the spindle reels them in, pulls them apart. So they line up in the middle, so when they get pulled apart, it's not like, if they just did it like this, they're like, okay, pull. Then all these would get tangled with each other, all this stuff would get messed up. It might work right most of the time, but a significant amount of the time, you'd have pieces getting tangled if they just started reeling right now before it was ready. Instead, they get them nice and lined up, they reel them in, and then that leads to the last one. Okay, so our last phase is telophase / cytokinesis. These are actually two different things, but they happen in sort of one smooth motion kind of. And what happens here is that everything in prophase happens in reverse. It's also, hey guys, you told me before you were talking about science. I don't think you are now. Okay, prophase in reverse, meaning that the nucleus reforms. So your nucleus reforms here. The chromosomes unwind because they're really only wound up for moving day, just like all your Christmas lights. Or whatever festival holiday lights you like to use. Because you don't use the Christmas lights all wrapped up, you put them on the tree. And so I guess you put the Christmas lights back up. I don't know what analogy you want to use, but the chromosomes unwind because that's how they do business. They do their thing with all of the chromatin exposed and unwound, so they can go back to being a cell. Because that's what they do. And then the third thing is that the spindle disappears. And so those are the three things. The spindle disappears because its job is done, right? It's pulled the chromosomes apart, half in each side, nucleus reforms, and then the cell goes through cytokinesis and reforms the cell just like it was before, except that now there are 2 of them. Now there's 2 of them. And so the idea is the cell has taken great pains to create 2 identical cells through the process of mitosis. And what we have now is just the same as what we had before, except that now we have 2 of them. Isn't that marvelous? Okay. Does anybody have any questions about this process? No? Alrighty. Okay, very good. Thank you very much for your time. And I have been Mr. Auerbach.